Hi everyone. Happy New Year. We've almost made it through the longest month of the year. Congratulations. This kicks off our first webinar of the year for MBF, and you'll be hearing a lot from us and from the MBF Bioscience team in the next coming months. We'll be holding monthly webinars, frequent office hours, and we'll be at all of your favorite workshops and conferences this year. Some introductions before we get started. I'm Maci Heel, and I'm joined today by my lovely co-host, Alissa Wilson. We are both on the product management team here at MBF, and we're stationed at our headquarters in Williston, Vermont. Today, we're going to be covering several topics surrounding neuron and vessel morphology, reconstruction, and analysis, taking a special look at cleared tissue imaging data.
We will start with a quick summary of the imaging done on the data that we're using today for the demonstration. Then we'll talk in depth about the data collection workflow, from visualization to morphological reconstruction to analyses, with tips on handling big data along the way. And though we'll be diving into software tools using cleared tissue as an example, the workflows and features we'll cover today are not limited to this modality. The toolset is specialized for a diverse set of inputs from various tissue processing and image acquisition techniques, from an entire cleared brain to sectioned tissue, from confocal, wide field, bright field, and even micro-CT. Ultimately, it can be utilized to answer a wide variety of research questions.
Definitely. We'll have a Q&A session at the end of the presentation, and we really want you to submit your questions as the presentation's happening. So go down to the bottom of your Zoom screen, open the Q&A button. It'll open that panel for you. Type your questions in, send them to us, and we'll get to them at the end. Now for the fun stuff, Alissa.
So the image data that we're going to explore is from a cleared transgenic mouse brain expressing GFP under control of the Thy1 promoter to visualize those motor and sensory neurons. And we also see tomato lectin in red showing the microvasculature. This brain was cleared using binary rapid tissue clearing, and this image was acquired using ClearScope, which is our light sheet theta microscope. The light sheet theta technology uses dual illumination pathways that are oblique to the specimen and detection pathways, enabling imaging of thick tissue specimens over a large lateral area at high optical resolutions while maintaining fast imaging speed, high imaging quality, and low photo bleaching. If you would like to learn more about this technology, please see the Expanding Your Research Possibilities with Light Sheet Theta Microscopy webinar on our YouTube channel.
Yeah, that one's a great listen. Our colleague, Peter Lang and Dr. Raju Tomar at Columbia University host that one, and it's definitely worth a watch. Great. So we've used our ClearScope to generate all this data. What can we do with all of it? And today we're going to talk about how Neurolucida 360 can be leveraged to reconstruct both the neuronal and the vascular morphology from both of those channels that Alissa was talking about. And you can use this tool set to answer unique and specific experimental questions. And we're really excited to show it to you today. And we're just excited in general that we've added the vessel tracing to our Neurolucida 360 software. There's a lot more in the brain than just neurons, and we added it so we can help our fellow researchers address really complex scientific questions about the brain and other organs that they're studying, use it to evaluate animal model efficacy, and just ultimately make new discoveries in your research.
And to do this, to begin, we offer some imaging processing features to set up your images for the most efficient reconstruction. The background subtraction filter will minimize the background in your images, bringing your objects of interest to the forefront and ready to reconstruct. This filter can be applied to a single stack or many using NL360's batch pipeline tool.
Also in that pipeline tool is the vessel filter. We understand that there are a lot of different ways that you can label vasculature in tissue samples. And we've seen that some of these methods can produce an image where the lumen of the vessel is unlabeled. And so the vasculature looks a little bit hollow. And what the vessel filter is going to do, it will programmatically identify the boundaries of the vessel and then fill the lumen in with higher intensity pixels to solidify those structures, making it easier to automatically reconstruct in an accurate way. There's also a morphological closing filter that's available with all of the other imaging filters in the software. So if your vessel labeling, instead of being hollow, maybe it looks punctate around the surface of the vessel. And what the closing filter will do is it will kind of even out the intensity across the surface, and again, make the objects more solid and able to reconstruct easily. We also have people who run, or researchers who use both the vessel and the closing filter together with much success.
And another new software application that is available to our researchers is Neurodeblur, a deconvolution software that can integrate seamlessly into your imaging and analysis workflow. Neurodeblur uses statistically optimal deconvolution image restoration methods to ensure that neuronal reconstructions are free from optical aberrations and are as accurate as possible.
Yeah, really excited about that one.
Mm-hmm. And now that we have acquired the whole brain and optimized the image, let's go through the steps to reconstruct the morphology within our region of interest. And today we're going to take a look at the cortex. And within the cortex, we will reconstruct the complex neurovasculature components within Neurolucida 360, which will result in a quantifiable model, which there's an example seen right here.
All right, let's do it. All right, I should be showing Neurolucida now. Okay, so now we're here in Neurolucida 360, and we have a large chunk of that whole brain image that Alissa was just showing us. And it's in our 3D environment in the software. Image data acquired with Lightsheet can be so big, as you know, and it's cumbersome to work with and to analyze. So we've integrated this new tool that I'm using here to virtually crop the image into a subvolume. And I'll adjust the size and the location of that. So we're just looking at a smaller region. That's all it will be loaded into memory at a time. So it really saves us computational and personnel time to investigate the automatic tracing parameters to use on this data. All right, so here we have just the red channel. I've isolated that using the channel panel selector on the left-hand side. And I'll open our vessel tracing dialog. There are 3 different tracing modes available for that vessel reconstruction. There's manual, user-guided, and automatic here. We will use the automatic today because there's a lot of vessels just in this small portion, and we want to do it fast. Right. So we're going to improve the tracing accuracy, and to do that, we'll adjust some settings to start. So first, the program will identify the foreground pixels that could easily be vessel candidates, and you'll see those are represented as seeds. And we can adjust the sensitivity of that and refine those seeds programmatically. And then finally, what we'll do is we'll adjust some more criteria to detect those vessels. We'll increase the sensitivity and remove traces shorter than a certain amount, and we'll trace those vessels. Here we are. One of the unique features of Neurolucida 360 is that it generates a 3D model, what you're seeing here on top of the image data. This data is stored in a separate file, and it allows us to do a robust set of analysis. And we can also easily edit parts of that model without having to redetect or rethreshold. I'll show you how to use these tools in just a little bit. But because the data is so dense, especially with this cerebrovasculature, we have added another tool to manage that big data, and it's called the partial projection tool. I'm using it now to virtually cut the data and then will be able to scroll through with just that slab of data showing it's around 122 microns at a time. So we can really inspect and make sure that things are accurate based on those parameters that we've chosen in the automatic tracing. So now I'm going to show you, I'm going to hide the image, and we're just going to take a look at the modeling data or the reconstruction. And in the vessel panel, there's within the editing tools is a new surface feature in Neurolucida 360. We're really excited about it. And what we can do is we can recolor the surfaces of those models, of those vessels. And in this case, I'm going to do this color by diameter. So I'm changing the color map that's going to be applied to the whole tracing, where pink will be the largest vessels, blue will be the smallest vessels. And you can see here, it's recolored just the areas around the model surface that are out of a certain diameter. And we can change how that color map is distributed, the way the gradient is, by adjusting the positions here in the customization panel, like so. But what we can also do, and this is even newer of a feature, is adjust the transparency of the bins. So if we want to take a look at just the widest diameter vessels, we can increase the transparency of the smallest bin and the medium bin to see just the areas there, like so, where those vessels are large or they've been dilated. And vice versa, we can take a look at just the smaller capillaries or arterioles and see those instead of the larger vessels. So I'm going to turn the colors back on, so you can easily switch back and forth from those two displays and make some really lovely images for presentations and for publications. You can change all the colors, customize that to your liking. So now let's take a look at the other channel. We're going to select the green channel here, and you can see what happened is the vessels in the red channel became transparent, and we can further increase or decrease that transparency using the trace opacity slider on the left-hand side. Because the data will be detected in the green channel, it will ignore everything that's been detected in the red channel. So you can do a lot of complicated tracing that is in the same space at a time and limit what you're seeing to make it easier for you. So let's take a look at the cell bodies. We will adjust some of the criteria for automatically detecting these somas. So I'm going to decrease that detector sensitivity, increase our search region to be a little bit larger than the size of the cells, and we'll detect the somas using this criteria. And that will result in cell bodies being detected throughout the image. We can go in here and we can remove somas. We can redetect with different parameters. We can also add somas that might have been missed by just a simple click to detect method, where we click and it will model those accordingly. Or you can redetect by changing the parameter. So maybe you want to do another round of detections with different sensitivity because you have a lot of cells that are like more dimly labeled. Then you can run an iterative detection. Now let's trace the tree. So we'll go to our tree panel and we'll run this user-guided type of tracing. So instead of everything, we're going to take a look at a single dendrite and trace along that projection. And you can see what happened as I was doing that is that the subvolume moved dynamically as we placed that last point. So you can imagine this subvolume tool would work really well if you were following like a long axonal projection across brain regions in your light sheet data or your other modalities without having to go in and out of that subvolume. But it's easy enough. As you can see, what I'm doing now is going back and I'm readjusting our subvolume so we can take another look at that region that we were originally interested in and trace the rest of those trees using automatic methods. So the parameters are going to look really similar from the automatic tree versus the automatic vessel detection. So the learning curve is really short once you've mastered one. In this case, we'll do the same seed detection to initiate the tracing. We'll do a little bit of refinement. And in this scenario, I'm going to use my cursor to remove some seeds from the bottom of the image here. That way we won't detect those cell bodies that we didn't trace to begin with. And you can do this too if you have some non-specific labeling that's occurring in your image, so it won't detect in those areas. And then lastly, again, we'll change the sensitivity and the tolerance and the branch connection criteria to automatically trace the trees here. And note that while this is running, those parameters, any of the parameters, whether it's tree, soma, spines, puncta, vessels, anything you're running automatically, those settings can be saved. And you can use those settings across your lab. You can use them for across sessions, and then you can use it across files of images in the batch pipeline as well. So here is the result of our tracing, and we can edit this. And so what I'll do is I'll go into our edit mode, and what that will do is we can look at this tracing point by point. This is available as well in the vessels, so you can adjust the points of the vessels too. But here you can see each tree is made up of several points, and we can change the size of these points. We can move these points. We can do a lot of different, oops, move that there. We can move it right back. And we can also remove many points at a time. So here you can see I'm zooming in on that area, and I will select multiple points and remove them. And then I will connect those two yellow branches that should be connected by dragging one point to another, releasing it, and that releases the tool, and now you have one contiguous tree. Now we'll exit the points mode, and we'll make the vessels opaque so we can see all of our neurons and vessels in one image, and like that. There we go. And don't forget that there is a movie mode as well. So if you're looking for ways to visualize and present your data, there's an embedded movie mode in the software too. I'm going to recolor all of the components here. So we'll color all of the trees that nice turquoise color and make the somas magenta, and then we'll go over to our vessels and color that. So you can see what I just did. Since I was in the green channel and I went to adjust my vessels, it says there are no vessels in that channel, which is a good thing. We're separating the channel data from one another so we can do a lot of analytics that span the different channels. And then we'll save that tracing, and we will open the data in Neurolucida Explorer.
Those new tools are great, and I'm especially excited for people to check out the surface feature. So now that we have all of this, so to get here, we utilized our large data management tools that allowed us to easily reconstruct the objects within our volume of interest. And so now we want to extract quantitative information about this morphology to answer our various experimental questions. And so we can do this using our many analyses in Neurolucida Explorer.
Great. Yeah, so Neurolucida Explorer is it's a companion software to Neurolucida 360 and our Neurolucida microscope software. And what we'll do is we'll bring just the tracing data here into the program. And we'll extract the metrics that we need. There are so many possibilities in this software, and it really depends on the type of research questions you have and what you're trying to answer. But we're constantly adding more analysis and visualizations at your request. We love to hear them from you. So we'll select the objects that we want to analyze. In this case, I'm going to select my vessels to start with. And in the Analyze ribbon, you'll see there are a section for vessel data analysis, neuron data analysis, region data. I'm going to start with this vessel network. And by clicking that icon, I have a new dialog where the program is requesting to report on some amount of metrics that we're interested in. In this case, what I'd like to take a look at to start is how often loops or anastomoses are occurring in the data and how long and what their volumes are. So we will select the anastomoses analysis here to investigate that. And I also want the program to bin all the vessel segments that I've selected by a specific diameter. Maybe we want to understand the distribution of capillaries to arterioles or venules in the region. So you'll see that we've instantaneously generated a report based on what we've selected. So the first here is the anastomoses. And you can see as I'm clicking on certain rows of the report, it's highlighting the corresponding loop in the software. We can also sort by this data too. So if we want to look at the anastomosis with the most nodes, I will sort that and click through those. And then for the bin segments, you can see again, it's doing the highlighting of everything that's within that 2 to 4 micron diameter bin. We can export all of this to Excel for more interrogation of the data as well. So we'll analyze now the somas and the dendrites and the tree structures in the data. So we'll go to the branch structure analysis. Again, there's so many things that you can choose here depending on what you're looking at, from spines to varicosities to, again, a global neuron summary, which I'll click here. This will show us how many cell bodies, their average area and volume. The same goes for the trees that have been selected as well. Endings, things like that. But there's more than just the structural analysis. There's also orientation. Here we can take a look. Maybe you're interested in the orientation of these trees. Here I'm using the polar histogram to visually describe that the overall direction. Maybe instead we want to compare the vessel or the neuronal field from one subject to another. In this case, we'll use a spatial analysis called convex hull. And what that will do is the program will generate a graphics of that convex hull, and it reports the volume and the surface area of the enclosed space that the vessels occupy. Or again, if you had like a single neuron, it would create a wrapper around that enclosed space. But maybe the total extent of the vessel field is too broad for your scientific questions. Let's take a look at if we were to have a region of interest, maybe a brain region. In this case, I've created some contours describing just an arbitrary region of interest to demonstrate here. But I'll select those regions of interest along the Z-plane, and I'll select my trees and my vessels, and we'll use close surface analysis. And what this will do is it will create a shell around my region of interest and report on just the vessels and the neurons that intersect or enclosed within that space. So that first report is going to show the network. So if I select my vessels here, you can see it's just, again, the graphics just highlighting what's inside that region of interest, that volume. And the same goes for the dendrites. So I'm really just scratching the surface here of analysis possibilities. And, you know, like I said before, building this to help researchers and meet your goals. So if there are things that you need, Alissa and I are ready to hear about them, and we're ready to work with our development team to add them in for you.
And in addition to reconstructing the neural and vascular morphology within your image, there are more tools to help you pull out even more information from your cleared tissue data.
Yeah.
So some of these, maybe perhaps you want to register a whole brain volume to a common coordinate framework, like the Allen Mouse Brain Atlas, perhaps, and then automatically delineate brain regions and even automatically count and compare cell population distribution across different subjects. Maybe you'd like to use stereology to find the estimated cell number, process length, size within your region of interest of that cleared or sectioned brain, or maybe some other organ samples. And you may be looking at things even smaller than cells. Perhaps you're interested in where synaptic proteins are distributed or colocalized. And we offer software solutions to allow you to explore your unique data and rapidly answer these questions. Check out our other webinars on our YouTube page to learn more, or contact us directly and you can see them in action.
Absolutely. So 2024, we're excited to see even more features added to our software for you. We really want to hear from you. Again, I sound like a broken record, but we really do want to hear from you about what you'd like to see. But just to give a little taste of what is coming up, we are employing methods of machine learning to analyze really dense axonal fields, also to analyze expansion microscopy data. We're adding more neurovascular specific analysis in our Neurolucida Explorer application, like I mentioned. We're focused on developing new tools and interfaces for detection of glia, as well as morphological reconstruction of glial cells. And then we're going to be releasing soon some enhancements to our puncta features, which is a really robust set of detection that you can do to do spatial distribution type analysis and colocalization analysis on puncta and cells. So, yeah. We're really excited for the year ahead, and we're excited to continue to support you. You all mean the world to us, and you're the reason why these products are so advanced at this point. It's because of your input and your feedback over the years, and we just want you to continue doing all of the groundbreaking work, and we want to help you do that. Yeah, thank you for joining while we get the Q&A set up. We just want a big shout out. Thank you.
Yes, thank you so much for joining us today. And a quick note, if you think of a question, or if we do run short on time during the Q&A, please contact us. You can also visit our forum page for more information, or please give our amazing support team a call. They can help you with all of your questions, both technical and scientific.
Yeah. We'll also be doing a new series of office hours with our product management team. As you might know, we have a really diverse set of products and solutions that we offer to researchers, and we are rolling out this new series so that you can come. Don't have to have an appointment, don't have to do any emails, just show up. We'll guide you through some of the new stuff, but ultimately we'll be there to answer your questions and listen to your feedback and feature requests that you have.
All right, I think we're ready for the Q&A session. Thanks everyone for listening so far and for sending in your questions. So we're done a little bit earlier than we thought, so we hopefully will get to all your questions. I have up on the screen now is a look to our upcoming webinars as well. So while this Q&A is happening, take a look at some of the things that MBF will be talking about in this webinar series in 2024.
Yeah.
All right.
Okay,
Let's see.
I can grab the first one. So One of the questions that came in is, can you reconstruct other things with Neurolucida 360 besides trees and vessels? And yes, you can reconstruct somas, spines, puncta, varicosities. You can also create contours, so making any anatomical boundaries that you need. And there's also markers that can be used to identify unique features. And another question had come in about, is Vesslucida included in Neurolucida 360? And yes, with the Ultra package, that is possible to be able to trace your neurons and vessels all in one software now, which is
Yeah, and
very exciting.
we'll still be offering Vesslucida 360 as well for those Vesslucida users, but we've ported the vessel tracing algorithms and analysis options into the Neurolucida 360 Ultra package. So if you're a current Neurolucida 360 owner, you want to give it a try, we're happy to set you up with a free trial. I think we'll talk a little bit more about that. We'll answer another question, actually, in if you can download the software. So we offer online demonstrations, free trials of the software, and for current customers as well, but new people too. Let's see here. Okay. So let's see. All right, we have a question about zooming in and out and handling the resolution, it sounds like, while you zoom in and out on the image to minimize computational burden. There's a feature that's somewhat recently been put into the software in the imaging panel. When you're in the 3D environment, there's an option called the visual density. And what you can do is you can increase the visual density to resolve to the fullest resolution of the section or the image that you're looking at in the 3D window. But if you need to reduce that computational burden and you don't need that high or full resolution image to be seen at the highest resolution, it will lower that res in memory at a time. Do you have anything to add to that one, Alissa?
I don't think so. Yeah. It can definitely come in handy, especially like we talked about with all this large image data.
Yep. All right. Let's see. Also have a question regarding detecting larger vessels and any tips for reconstructing those larger vessels. And there's a few things I would suggest. It really depends on the data. So I'd love to take a closer look at your specific data to the researcher who asked this question. But off the top of my head, I would check out the typical process width setting. It's available in the user guided mode, but it will carry over into the automatic mode. And you can input a micron distance of the diameter or the width of those vessels that you are looking at, and that will help guide the algorithm to target those larger vessels. I also find voxel scooping. That's one of the vessel tracing algorithms available to use, again, in both the user guided and the automatic mode. And the voxel scooping algorithm tends to work pretty well for those larger caliber vessels. And like Alissa and I were discussing earlier in the webinar, there are some filters to help kind of prepare your image data for the reconstruction. And so depending on your data, it might be helpful to run those through either the vessel and or the closing filter to solidify those larger vessels. And then, sorry, part of that question is how do you handle vessels that have different levels of signal? And for that, depending if they're in the same field of view, I would run two separate detections. So you can run automatic detections using one set of parameters, detect those, let's say the things that are closer to the foreground, and then run another set of parameters to pick up the vessels that are closer to the background using maybe a higher intensity. if they're in different areas of the image, you might want to use the subvolume tool to isolate certain regions that have fainter signal and use higher sensitivities or different connection criteria for those areas.
And then we also have a couple questions about the largest file size that can be processed, and a couple about like, again, the resolution of the data. So what is the minimum resolution needed? And PC requirements. So
yeah,
kind of putting those together to answer. And obviously, please contact us, and we can take a deeper dive into your image data and your PC and make any suggestions. But so I guess the quick answer is there's no specific limit, but it is dependent on your computer and the complexity of your data. So the more markers that you have, the more objects that you have, the more image data, you're going to need the computer power to be able to visualize that. And I think
we can definitely make specific recommendations too that will help guide you in that process. And again, we do have a eval trial demo program. So if you have a system, you're really interested if it's going to, how it's going to perform, we can work with you on getting it set up and trying it there and seeing where, what priorities would need to be made to upgrade the hardware on the computer, or, really diving into some of the tools we showed earlier with the subvolume and the projection tools to limit that.
To kind of cover the resolution of your data, again, that it's going to depend on what you're looking at. If you're looking at those large vessels, you don't need the resolution as someone trying to look at something tiny like spines or other like small objects. So again, that will really depend on what you're looking at, but you definitely want the resolution so that as you're moving through your stack that you can identify changes and be able to easily see those objects.
Absolutely. Okay, let's see. All right, looks like there's a question about measuring glia and vessel colocalization, or astrocytic end feet coverage of vessels. And that goes back to what Alissa had mentioned at the very beginning of the question and answer session is there are other tools in the Neurolucida 360 software that we didn't go into today, but that can be utilized to answer questions like that, specifically the puncta tool. And the puncta tool can be used to detect various punctate objects of different shapes and sizes. And so that tool specifically has a feature to detect based on proximity. So you can detect puncta in one channel by proximity to vessels in another channel, or to a cell body. So you can look at intracellular proteins, or how synapses are forming and things like that. So there's a lot of things you can do using a combination of those tools to answer those questions. All right.
Oh, and then, okay, so we also had another question. Does this approach also work for a series of slide scanned images, TIFF image specifically? And yes, you can open the series as a stack to do the reconstruction. This will work the best if you have, a smaller Z-step, but again, depending on what you want to reconstruct. And also if your images aren't aligned, so if you go to open them in as a stack and they're not aligned, we also have BrainMaker, Tissue Maker software, and that will align the sections for you, and then you can save that as a stack and bring it into Neurolucida 360's 3D window to begin your reconstruction. So
that reminds me, not a question, but a point that the Neurolucida 360 software does accept many imaging modality inputs. Yeah. So it's not TIFF only. You can use a wide variety of proprietary formats, ND2, LIF, JPEG 2000. The list is quite long. Someone's asked if this can be used to trace neuronal growth in 2D culture, and absolutely. Just like we support many file formats, we support data from different modalities, both 3D and 2D data. So the tracing algorithms will also use the information from just a single plane in addition to a 3D stack.
And then are all the question about new tools and options available on the older versions of Neurolucida, and some of the things that we went over today are available there, but anything that is newer is only available in our the newer software. So happy to, yeah, please contact us and we can get you upgraded.
Yep. And on that note, and something we should have said way earlier, is that we will be releasing a new version of Neurolucida 360 this week. I can't believe we forgot.
Yes.
So that will have all the features that we showed in the webinar.
Yes, definitely. Yes.
And more. All right. Okay, let's see. Question here, you mentioned some pre-processing techniques in the beginning of the webinar, like background subtraction, vessel filtering, neurodeplorer. How do we use these features? So the filters, background subtraction, vessel closing, there's a filter in there for Golgi data, for in vivo, to create max projections. All of those are available in the batch pipeline, and that is in the imaging, the workspace ribbon in the software. So that batch pipeline, we have a webinar that we put out last year about all of those tools. So I definitely recommend giving that a listen or contacting us, but that goes through the steps to get there. And the nice part about it is that you can run those filters across many images at once in that batch filter. Or you can use it to find the correct settings for your images. There's a few different settings for each of the filters that you can adjust for your specific data. Neurodeplorer is a separate software. It's not included in the Neurolucida 360 package, and that is available as a separate download, and we can also get you started with that if you're curious. Some questions about the Explorer functionality. Let's see here. Function to calculate the nearest distance between an axon and a vessel. We do have a proximity analysis available, but we'd love to hear more about your specific needs for that. Like we said in the webinar, we just put the vessel analysis together in with the neuron analysis in Neurolucida Explorer, and we're really hoping to expand that tool set so you can do the specific investigation you need to do. So I would love to follow up with you about those specific needs and what kind of data you'd like reported there. But we can calculate the distances between those objects. And then does the analysis give a distribution of the diameters of different vascular segments and their lengths? We also report the diameters of the segments themselves, so the average diameter of between two nodes, but also specifically at each point along the vessel, and the length of those segments too. So specific segments, whole networks, and down to the points. There's a question for how it performs for segmenting noisy CT and microscopy data, since the examples we showed were clean for our demonstration purposes, but all the algorithms can be tailored to your data. So there's certain criteria that I adjusted here in the demonstration, but there's even more advanced settings that you can adjust depending on the quality of the image data to address maybe more complex situations.
And the image that we use today, we did use the background subtraction filter.
Yes, that too.
To help as well. So
yes, yep, absolutely. There's a question about how the software handles detection of multiple dendrites branching off a soma. This one was very simple, where we saw single branches emanating from the soma. But we handled that a little uniquely, where the somas are distinct objects from the trees or the vessels or the puncta or the spines. And that allows you to do a robust set of analysis. But it also means you can have branching structures or more than one tree emanating from a soma at a given time. The software doesn't make any discrimination one way or the other. And then the second part of that question is when there's more dense labeling of cells as opposed to what you saw, which was the Thy1 GFP. And again, we really try to make the software flexible for different types of inputs and different complexities. So again, using those settings and to improve the accuracy of the tracing, or even using user-guided methods for really complicated areas is possible.
And there was also a question about the tree tracing mode, and if it's possible to decrease the distance between the beads of the trace. So in automatic, when you are setting up your settings, there is a seeds tab, and you can make that. There are 3 options, radio buttons, that can make them more packed or more like more dense or more sparse. And so yes, you are able to do that when setting up your trace, that automatic tracing.
Yeah, and even for the user guided, so that interactive tracing method, I find that the different algorithms, so we have voxel scooping, directional kernels, and Reber's crawl available in the software for the user guided tracing. And since those algorithms work a little bit differently to detect and generate those seed points, I find directional kernels, for instance, produces more points along the center line than, for instance, voxel scooping.
Yeah.
So if you need that denser tracing, I would explore those options.
Okay.
There's a question.
Sorry, you guys are asking some really great questions.
Yes.
I just want to make sure that we get them all.
I know the list is quite long. There's one here about the nodes. I just lost it. About the number of nodes, and I think in the example I had filtered the report by number of nodes, so it looked like all of the nodes, number of nodes for each of the anastomoses were 15, but it was actually many of them had 15 nodes, but not all of them had 15 nodes. So I just clarify that. But that would be a way to measure the vessel branching through the branch structure report for vessels and looking at the number of nodes in the selected in the selection. All right. Let's see here. All right. I think there's a lot of pricing questions, upgrade questions, and I encourage you to reach out. And I will put my email in the chat as well as the MBA or the info email box. Let's see if I can do that here, and we can talk about those options that you have. Oh, looks like I can't send it. We will follow up with a large email to everybody who had those questions. Sorry.
Yeah, and also about like any trials and demos. There was a few about that too. So, and the trial and demo, I think there was a couple questions about the data. So we are happy to provide a link so that you can send us your data and we can do the demo and the eval on your data too.
Yeah,
so
rather than the example images that we have.
exactly.
Yeah, we definitely know it's different challenges for each data set. And the general email is info@mbfbioscience.com. So you'll get to the right person if you go there.
Right. Oh, there's a question that came in about vessel branching. If we can analyze the vessel branching similar that we can for dendrites, and yes, there's shoal for vessels as well.
Yeah, the shoal, you can place a marker where you want the center of the shoal.
Right. Yeah.
So there's some custom options you can do there depending on how you want to analyze that.
Yeah, because typically with dendrites, the shoal is centered by the soma. So for a vessel, you can use a marker to do that.
Is it possible to do Atlas registration 2D and 3D in Neurolucida 360? Right now, all of that technology we have for registration of 2D or 3D images, whether they're serial section or from a light sheet, all that lives in our NeuroInfo software. And I highly recommend giving that a try if you're doing that type of investigation. It is possible to, if you are interested, you have light sheet data that you're using in NeuroInfo and you've registered to an atlas and done the automatic delineation of brain regions, of your regions of interest or all of them, you can bring those contours and the image data into Neurolucida 360 and do some morphological reconstruction and then use like I showed, the closed surface analysis in Neurolucida Explorer to extract metrics about what's interfacing within that one or intersecting and enclosed in that volume. And again, we'd love to hear if you are interested in doing something like that. We can set you up with a nice workflow that interfaces with both of those software.
Definitely.
So also another BrainMaker question about using BrainMaker on serial sections that have been Z-stacked on a confocal microscope. There might be, I wonder if it's, I guess there's two parts to the question, depending if BrainMaker itself will do the stacking of those serial sections. NeuroInfo, however, as BrainMaker is part of that, NeuroInfo will work on serial sections that have been realigned into a Z-stack, so you can do all of your Atlas registration there.
Oh, and then there was a question about nuclear detection by region. In cleared whole brain images, is it better to use NeuroInfo or Stereo Investigator? What is the benefit of using one instead of the other? So
it's a loaded question.
Yes. And we can definitely follow up too about your specific, but I mean, I guess the general answer is NeuroInfo, you are using the cell detection workflow. So you're just kind of getting, a heat map of what is going on within your image data. So, you'll be able to see like concentrated markers. Whereas with Stereo Investigator, you are getting using stereology and getting an estimate of the total number of cells within that region. So, our cell detection is meant to kind of give you a broad picture of what's going on and say, oh, looks like there's a lot of activity over here. And then you can narrow in and say, okay, this region I want to get a cell count. And that's where stereology is going to come in and give you that estimate of those cells.
Yeah, unbiased and
yes, yeah, yep. And then, yeah, so that's kind of a very brief overview of the differences, and we'd be happy to chat more about what you're doing and give suggestions. So
yeah, we have several neuroscientists on staff as part of our tech support team and our product management team. And so especially if you're interested in exploring a certain question and you're considering using stereology versus an automatic cell detection method, we would be happy to consult with you.
Yeah, definitely.
I think there was one more here. Did I just lose it? Let's see. There was a question about other analysis software failing to detect large vessels whose walls are labeled and not the lumen. And again, that's really or one of the reasons why we produce the vessel filter is to address that specific challenge where you have a difficulty targeting the vessels because they're not solidly filled. And that vessel filter in the batch pipeline has been really helpful in those scenarios. We've also, I guess, in addition to that, and there is a user-guided method that we have added as well. So that interactive tracing, where we trained a machine learning model to also address that issue. So it will kind of interactively close or fill in the lumen as it's tracing or recognize that it should be filled. And using machine learning, we are hoping to make that even easier for you to do on that data, to do accurate tracing. And that machine learning model is available in the latest release of Neurolucida 360. It's called CNN sectioning. So you'll see that in the drop down where you see your user guided and your, where you see your directional kernels and voxel scooping and Raper's crawl methods.
And another note, if you do want to share this with other members of your lab or if everyone wasn't available, this will be up on our YouTube channel, so everyone will be able to rewatch.
Well, that was a great Q&A session. We're at 1 o'clock Eastern time, so we will wrap up here, and we really hope you enjoyed the presentation, and we are really excited to hear more from you at the office hours and in the webinars and through email. So have a great day.
Yeah, have a great day, everyone. Thank you.